Hi, thanks for stopping by today. My name is Heather, and I'm going to tell you about the human infant brain. Virtually all healthy human adults have an area in the ventral temporal cortex that responds more to faces than to objects. In fact, the fusiform face area responds more to faces than any other stimulus category. The FFA is not the only category selective region. The occipital face area is also selective for faces, while the parahippocampal face area and the occipital face area are selective for scenes, and the fusiform body area and the extrastriate body area are selective for bodies. A fundamental question is, how does selectivity develop? Many studies have shown that category selective areas increase in size and selectivity well into adolescence, suggesting a long, protracted period of development. But a lot of change happens between infancy and childhood, and the human brain is growing a ton. We actually know very little about the infant brain. What is the functional profile of an infant brain? From one study, we know the infant brain has face and scene responses in approximately the same areas found in adults. But the authors did not observe a face response that was larger than the object response, a signature of face selectivity. Similarly, the observed scene response was not greater than objects. Thus, category selectivity was not observed in the human ventral temporal cortex of infants. But is this really the right answer? Today, I'll first walk you through an experiment I conducted to replicate infant face and scene responses. Then, I'll tell you about some innovations we did to improve infant fMRI data collection. Finally, I'll walk you through the results of a second experiment by talking about category selectivity specifically. All right, first, the replication. For those of you that are not familiar with infant fMRI, we first swaddle the infant and place them in a special infant coil. We use a sinusoidal EPI that is quiet and safe for infants' ears. An adult goes into the scanner with the infant, while the baby watches movies in a mirror over their eyes. As you can see, this baby is moving quite a bit. Babies watched videos of faces, bodies, objects, and scenes. To hold baby's attention, we also had a baseline block that consisted of colorful abstract videos. The movies played continuously as long as the baby was content and awake. For this study, we recruited 48 infants and collected a total of about 35 hours of data. We removed high motion segments and created subruns, which left us with about 9 hours of low motion data from 26 subjects. But only 16 of these subjects had enough data for their ROI analysis, leaving us with a total of about 7 hours of data from infants that were 3 to 9 months old. After all of that work, what did we find? In the group map, there are face responses in the STS and scene responses in the ventral temporal cortex. But what we really want to know is if infants have face and scene selectivity. The resolution of our data was degraded, so we used a parcel that covered the entire ventral temporal cortex. We selected the top 5% of voxels that responded more to faces than objects, and the top 5% of voxels that responded more to scenes than objects. On this plot, I will show you the average magnitude response on the y-axis. In independent data, for this group of 16 infants, here is the face response in purple. As you can see, the response to faces is greater than the response to bodies in pink, objects in blue, and scenes in green. But the difference is not significant. What about scenes? In independent data, the scene response is not greater than the response to all other categories. Is our failure to observe a category selective response due to a true difference between infants and adults, or is it because of a difference in data quality? I'm sure you can all imagine that infants are not exactly the best subjects. They move a lot, often withdraw consent by crying, or needing food or a diaper change, or if they are happy, content, and comfortable, then they fall asleep. On top of that, we used a sinusoidal EPI, which distorted the data so much you can barely even tell these are brains. Are these data the best we can get from awake infants? In a large group of infants, we replicated face and scene preferences, but we failed to find category selectivity for either faces or scenes. I don't know about you, but since these data are so noisy, I personally want to know if this difference between adults and infants is a true difference, or simply a result of noisy, distorted data. As such, our next steps were innovations to try and increase the quality of the data we collected. First, I designed a set of scanner-safe headphones that allow infants to listen to music while attenuating the noise of the scanner. This allowed us to use a standard EPI sequence without harming infants' ears. Then, in collaboration with Boris Kell, we designed a new infant head coil. This one maximizes infant comfort by having cutouts that accommodate headphones and support a variety of infant head sizes. The frontal coil then flexibly closes around the infant's head and is held in place by inflatable pillows. So, did the new headphones and coil make a difference? As a reminder, this is what the data looked like with the old coil. Compared to an adult 32-channel coil, the new infant coil increases SNR by 25%. But will it make a difference with awake babies? Here's the data from the new coil. It actually kind of looks like a brain. Our next innovation was with the stimuli. We kept the same 4 categories, but instead of 4 conditions, we had 12. 3 face conditions, 2 body conditions, a condition with hands and objects, 3 object conditions, and 3 scene conditions. We organized the stimuli in this way in order to determine the representational space of infant cortex. Finally, to enhance infant attention, we added still images to the blocks of dynamic stimuli. Each movie played for 2.7 seconds and was followed by a 300 millisecond presentation of a still image. All right, so for experiment 2, we recruited 32 infants and collected about 14 hours of data. Similar to the last experiment, we were able to collect enough data for the group random effects analysis from about 50% of the subjects, which amounted to about 7 hours of data. The new coil allowed us to keep nearly 50% of the data, whereas with the old coil, we only kept about 25%. For the ROI analysis, we still had 16 subjects with about 6 hours of data. This time, however, we retained about 45% of the data, whereas with the old coil, we only kept about 20%. So not only do we have better data from the new coil, but we also retain more of the data that we collected. The babies in this data set were also 3 to 9 months old. So now we have new headphones, a new coil, and better data. Will infants still lack a category selective response? All right, first, do infants have a face selective OFA or FFA? When faces were contrasted with objects, we did not observe a face patch around the area of the OFA. However, there was a face patch in the ventral temporal cortex in approximately the same area as the FFA. For the ROI analysis, we used small group-constrained parsers generated from adults to localize specific regions. I've projected the OFA and FFA parcels here onto an infant brain. On the y-axis is the average response magnitude. Here is the OFA face response in purple. It is higher than the response to bodies in pink, objects in blue, and scenes in green. The only significant difference, though, is between faces and scenes. So, in infant OFA, we did not observe a face selective response. What about the FFA? Here is the response to faces. As you can see, it is greater than the response to all other conditions, indicating infants do have a face selective FFA. Now let's do the same thing for scenes. Do infants have a scene selective response in either the OPA or PPA? In the group random effects analysis, we did not observe a scene patch around the area of the OPA when scenes were contrasted with objects. However, in the VTC, there was a scene patch in approximately the same area as the PPA. Is this response selective? In the infant OPA, the response to scenes is not selective. However, in the infant PPA, the response to scenes is significantly greater than the response to all other categories, indicating that infants do have a scene-selective PPA. All right, finally, we sought to determine if infants had a selective response to bodies in either the EBA or the FBA. In a group random effects analysis, there is a body patch in the left occipital cortex in approximately the same area as the extrastriate body area. There is not, however, an analogous response at the site of the adult fusiform body area. Functional region of interest analyses corroborate these results and demonstrate that the body response in the infant EBA is selective, while the body response in the infant FBA is not. All right, as a wrap up, today I told you about category selective responses in the infant brain. In the first study, I told you about how we replicated previous failures to find face and scene selectivity. I then told you about a variety of innovations we made in order to improve the quality of infant fMRI data. Finally, with these higher resolution data, I described a series of results that demonstrate infant FFA is selective for faces, infant PPA is selective for scenes, and infant EBA is selective for bodies. To be clear, face and scene selectivity was not observed previously because of the low quality of the data. Innovations in fMRI data collected resulted in better data, and we observed category selective responses in the infant brain as a result. A puzzle raised by these data are, why are some regions selective in infancy while others are not? Further, many people have failed to find category selective responses in children, yet we find selectivity in infants as young as 4 months. What can account for this difference? And with that, I'll say thank you. Thank you for your attention, and thank you to all of the people that helped me, all of the babies and families that participated, and my funders.